Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and we are going to do a quick review for Exam 2. Again, like the review video for Exam 1, we won't have time to look at everything on all the concepts and go over all the concepts, but we do have time to go over some of the things you should definitely focus on for the exam. Again, this is not a totally comprehensive review. You should study every slide. However, this is at least focusing on some of the key points that you should definitely know. Okay, so let's go ahead and get started. Let me fix the screen here. All right, so starting with chapter 6, we're looking at viruses. You should definitely know what a virus is, and that you have the DNA part, and then you've got the capsid part, and that constitutes the nucleocapsid, right? So you've got the nucleic acid, and that nucleic acid could be either DNA or RNA based, and then you've got the capsid, and together that constitutes the virus particle or nucleocapsid. Again, the capsid is the protein shell around the virus, and then you've got the nucleic acid, which means the genome. Remember this for the exam too, that viruses can have a multitude of different genomes. They could have DNA genomes, or they could have RNA genomes. They could have single-strand RNA genomes, double-strand RNA genomes. They could have single-strand DNA genomes, double-strand DNA genomes. So you should know that viruses have a diverse array of different genomes that they could possess. And the simplest viruses are simply just nucleocapsids, along with obviously other proteins as well, like enzyme proteins and such. But in essence, they are nucleocapsids. Now you could have the nucleocapsid, and that's naked, which means there is no envelope around the nucleocapsid, or you could have an enveloped virus, and that means that there's an envelope present. And you need to know for the exam that where does the envelope come from? Do viruses make their own envelope? The answer is no, they don't. They actually borrow the envelope from their previous host. So during budding, you know, when the virus buds out from the cell, during budding, the virus picks up the membranes of the host, like the plasma membrane of the host, for instance. Okay, then you've got spikes. You should know what spikes are. These are proteins embedded in the envelope of a virus, and they help the virus to attach to surfaces. And it's not just in the envelope. It could be on the surface of naked viruses, on the surface of enveloped viruses, but it's these spike proteins that actually allow for adsorption, which means the first phase of a viral infection, where the virus must attach to your cells, right? And I'm just skipping stuff. Again, just because I'm skipping stuff does not mean that it's not important. Definitely know all this material. I'm just skipping stuff so this video is not 3 hours long. So capsomeres, these are the protein subunits of the viral capsid. Let me get to something else. Oh yes, obviously the envelope, like I said, the envelope is very important. Not all viruses have an envelope. The ones that do not are called naked viruses. And remember, where does the envelope come from? It comes from the membrane of the host, or sometimes multiple membranes of the host. The genome can be both DNA or RNA. Remember, viruses don't have just one kind of genome like we do. Us organisms, we have only one option for genome. Think about this. If you're a living organism meaning, a bacteria, archaea, eukarya, human, whatever, we all have only double-stranded DNA genomes. Does that make sense? Whereas viruses can have a multitude of different genome types. You could even have positive sense RNA or positive strand RNA or negative strand RNA. What's the difference? Positive strand RNA can serve as mRNA, whereas negative strand RNA, negative sense RNA, cannot serve as mRNA. Okay, these are just the different types of viral genomes. Okay, again, I'm skipping stuff, but not because it's not important. Oh yes, you should know the stages here, the phases of a viral infection. Remember adsorption, and what's important for absorption, remember? Spike proteins. Penetration, all viruses have to do the penetration. That means they have to go inside of the cell. Uncoating, synthesis, assembly, and release from the host cell. And remember, during release, that's where enveloped viruses pick up their envelope. Now, I do want you to know for the exam that all viruses have to penetrate the cell, but not all viruses have to uncoat. You know why? Because bacteriophage, for instance, the T4 phage, the capsid part of the virus stays outside of the cell, and then it injects the DNA part of the virus into the cell. So there's no actual uncoating that happens. The only thing that penetrates the cell is the DNA and not the capsid portion of the virus. Okay, and you should know that you should know that enveloped viruses are animal viruses only. Okay, so only animal viruses can have the potential. Not all animal viruses have an envelope, but only animal viruses can. Again, I told you earlier with penetration step, all viruses conduct this step. And remember, I told you with budding, that's where enveloped viruses pick up their envelope. Great, let's keep going. We're making good progress here. See, this is what I was talking about earlier. This is the bacteriophage. And this is the capsid portion of the bacteriophage. And look what it's doing. It's injecting its viral nucleic acid, its double-strand DNA in this case. It's injecting that double-strand DNA into the cell. That's where the penetration is happening, but the capsid portion is staying outside. So there would not be any uncoating that occurs in this particular situation. All right, and then we talked about the lytic versus lysogenic state of bacteria, or bacteriophage, I should say. You should know how lysogeny works. Remember, lysogeny, you have the bacteria can either incorporate itself into the host genome as a prophage, right? And then it can switch to the lytic phase, which means that the progenome can excise itself, or it can actually cause viral replication and lysis of the bacterial cells. So these are called temperate phage. Temperate phage are the ones that can do both lysogeny and lytic cycles. So we should know what a prophage is. That's the portion of the viral genome that incorporates itself into the host genome. Okay. And you should know that lysogenic conversion, all of this lysogeny and this the temperate phage, these are terms that only refer to bacteriophage, not animal viruses. What else do we have to emphasize? Plaques. You should know that when we're talking about plaques, you should know we're talking about these areas of clearing where viruses have lysed the cells. So if you have like a monolayer on a petri dish and the temperate phages have switch to the lytic cycle, well, that will cause all those cells in that colony to lyse, and you'll get these clearings in the agar, and that is called a plaque. Okay, so you should know that plaques are clear, well-defined patches in the cell sheet due to the lysis of those cells during the lytic portion of a viral event. See these plaques? You see, this is a petri dish, and if you look closely, you got these clearings. See, all the fuzzy stuff, all the opaque stuff is bacteria monolayer, right? And where you see these dots, those are actually clearings of no bacteria on the monolayer. That was caused by a lytic event. That was caused by lysis of the cells due to the temperate phage switching to the lytic stage from the lysogenic stage. You should know for the exam what are prions. You should also know what are viroids. Definitely understand viroids as well. Okay. Let's see, we may be close to done with this chapter. We are done with this chapter. Let's take a quick break time, and we'll come back with the next chapter too, and what I want you to emphasize on. All right, guys, be right back after a break. Hey everyone, welcome back. Let's carry on with our review. Chapter 7, Microbial Nutrition, Ecology, and Growth. Obviously, know these concepts here that I'm flipping through. I'm going to flip through until I find something that I want you guys to focus on. These terms, please understand these terms. What is an autotroph? What is a heterotroph? Remember, that's where they obtain their carbon from. Also with the energy source, what's a chemotroph, a phototroph, a lithotroph? Please know what these terms mean for the exam. Again, as I just mentioned, phototroph versus chemotroph, you should know that most organisms do not live and survive in habitats similar to the human body condition. So what's the human body condition? You know what it is. It's 32 or 37 degrees C, 98 degrees Fahrenheit. That would be a mesophil with the pH of the body and the salinity of the body. Most organisms do not thrive in this environment. There are psychrophils that enjoy cold temperatures, thermophiles that enjoy hot temperatures. There are anaerobic bacterium that exist where there's no oxygen at all. There are all kinds of aquatic organisms as well. So for the exam, please know that most organisms do not live on the human body. And just fun fact for you, it has to be an organism that prefers the human body conditions. Those are the organisms that cause human diseases, right? So like a hyperthermophile isn't going to cause a human disease. It's not going to cause an infection on your body because a hyperthermophile can't even grow on the conditions of your body. What else? Obviously, you know what pathogens are. Understand the concept of diffusion, please. You know, substances move from a concentration of high, from an area of high concentration to an area of low concentration, and this is a spontaneous process. And you should know that osmosis is a form of diffusion. Osmosis is movement of water, or I should say the diffusion of water through a selectively permeable membrane. You should know that for the exam. Now, definitely know for the exam, tonicity, the concept of tonicity. What's happening with isotonic conditions, hypertonic conditions, hypotonic conditions? What would I want you to know? If the water concentration, or you could think of it as solute, if I'm talking about solute concentration, I'm talking about what's dissolved in in the water, for instance. If I'm talking about the solvent, I'm talking about the water. Usually with biology, it's water, right? Water is the solvent. So if the water concentration is equal inside and outside of the cell, then there's no net osmosis happening across the cell membrane. The amount of water osmosing out is the amount of water coming in, so there's no net flow of water. Why? Because the solute and solvent concentrations inside and outside of the cell are equal, so that's called an isotonic condition. Hypotonic condition, I want you guys to understand hypo sounds like what? Here's the easy way of remembering this. Hypo sounds like hippo, right? Hippo, like a hippopotamus, you know, and hippopotamuses they look all swollen up, right? So what you should know is that in a hypotonic condition, this means that water is rushing in to the cell, causing the cell to swell up like a hippo. You know, that helps you remember it. And why would that happen? Well, the water concentration outside must be higher than the water concentration inside, because so water is going to move through osmosis from the area of high water concentration, which is outside of the cell, to the area of low water concentration, which would be inside of the cell. Okay, and that's if we're talking about water concentration. What would the solute concentration be in a hypotonic condition? In the same exact example, it's the opposite. Does that make sense? The solute, let's say the solute is salt. Salt would be higher concentration inside of the cell, and salt would be lower concentration outside of the cell. Does that make sense? The water is going to move from where water is high concentration to where water is low concentration. The salt isn't moving, but the salt has the opposite concentration level, right? It's higher inside than outside. The water is higher outside than inside. So water's the only thing that can move. Water moves into the cell swells, the cell explodes. Okay, so that's a hypotonic condition. Now, what's a hypertonic condition? Exactly the opposite. Hypertonic conditions cause water to leave the cell, and the cell will shrivel up, right? The cell will shrivel up. Why would this happen? The water concentration is higher inside than outside, but the salt would be higher outside than inside. Does that make sense? That's what's going on. Remember, the salt isn't moving. In these tonicity problems, the water is moving. Okay, so please know that for the exam. Which way would the water move? And, you know, where is the solute concentration, high or low? What will happen to the cell? Will the cell lyse? In a hypotonic environment, the cell can lyse if it doesn't have a cell wall, right? In a hypertonic environment, the cell will shrivel up. In an isotonic environment, the cell, you know, there's little stress. You see this? There's little to no stress on the cells. Okay, obviously know the concepts of diffusion, active transport. What do I want you to know? I want you to know that phagocytosis, you know, the process of bringing in food, for instance, or bringing something in through the plasma membrane, that's an active process. That's active transport. Whereas what's diffusion? What's osmosis? Is osmosis active or passive transport? That would be passive. That's right. That's passive transport. You should understand the cardinal temperatures: minimum, maximum, optimum. And one thing I want you to know is that at minimum temperature and below, the cell doesn't necessarily die. It just can't move fast, and it can't divide fast, right? So because the molecules slow down. Does that make sense? Minimum temperature doesn't necessarily kill the organism. It just slows it down to where it's almost stopped metabolism. Think about it this way. If you put food in the fridge, it'll last a long time without spoiling, but eventually it will spoil, and that's because the cells are dividing very slowly. It's not like they're dying when you're putting the food in the fridge. The bacteria is still there and it's still alive. Now, conversely, if you pass the optimum temperature, usually you do kill the bacterium, unless it has endospores or it's a particularly heat-resistant type of bacterium. Okay, what else? Yeah, you see, below the minimum temperature, activities are limited, like metabolism, growth, cell division is limited. It's not necessarily that the cells are dying at this minimum temperature. You should know the types of bacterium that grow in different temperatures. Psychrophiles, you should know this for the exam. Psychrophiles are the ones that can grow in very cold conditions. Then you've got psychotrophs, which can grow in slightly warmer conditions. And then mesophiles, you should know that mesophiles are the ones that grow about at human temperature. You know, humans' bodies are at about 37 degrees C. 98 degrees F, and those are mesophiles. So most of the bacterium, you guys, a lot of you are going to move on to become healthcare professionals, right? Like dental hygienists, nurses, etc. You should know that a mesophile, those are the ones that are going to cause gum disease. Those are the ones that are going to cause the infections, the skin infections, the gut infections, the, you know, all the different pathogens that infect the human body. Those are mesophiles, right? Then you got the thermophiles, which can exist in hot temperatures, and the extreme thermophiles that can exist even in boiling temperatures sometimes. Okay. Oxygen sensitivity. I don't know if you need to know this, but catalase and superoxide dismutase, these are enzymes that bacterium need in order to live where there's oxygen. Oxygen is very oxidative. Oxygen destroys DNA, and it really is harsh. So unless an organism has catalase, peroxidase, superoxide dismutase, these enzymes allow organisms to grow where there are, you know, ROS, like a reactive oxygen species. I want you to know that catalase is actually the enzyme that deals with the toxic hydrogen peroxide molecules, okay? Hydrogen peroxide is broken down by catalase. You may or may not need to know that for the exam. So you should know the terms aerobe, facultative, anaerobe, microaerophile, anaerobe, aerobe. pH, acidophile versus alkalinophile, osmophile. Oh, you should know barophiles. So barophiles are organisms that thrive in high pressure conditions, such as at the bottom of the ocean. Okay, barophile. Okay, we should understand symbiosis and the examples of symbiosis, mutualism, commensalism. Definitely know the terms commensalism, parasitism. Please know those for the exam. Binary fission. Oh, with binary fission, I do want you to understand for the exam how growth works. You know, when one cell becomes 2, becomes 4, etc., etc. What if I asked you on the exam, how many cells would you end up with if you started with 1 cell and there were 4 doublings? Could you answer that on the exam? Think about that. If you had 1 cell and that cell doubled 4 times. Now you could just think about it logically. You could say, well, if 1 becomes 2, those 2 divide to give you 4. And you could think of it that way. Or you could use the simple equation of exponential representation here. You could do to the power. So what was my question? I said if a cell divides 4 times, you see this? If a cell divides 4 times, then the equation for that would be 2 to the power of 4. Does that make sense? 2 to the power of 4. So you could either answer 16 by thinking about it mentally, or you could just plug into 2 to the 4, and that would give you 16 experimentally. Either way, you would get the answer correct, right? Does that make sense? So please know how exponential growth works, doubling times work, and if I were to ask you a similar question on the exam, be prepared to answer. Please know how batch culture works. You've got the lag phase, exponential growth phase, also known as the logarithmic growth phase, stationary phase, and then death phase. A death phase is also known as senescence. And then kind of understand what happens during each phase. Lag phase, exponential growth phase, stationary phase. One thing you should know, you may want to know this for the exam, is that cells can stop growing or grow very slowly, but they're not necessarily dead. Even if they're non-modal and not growing, that doesn't necessarily mean that a bacterial cell is dead. Okay, and then during death phase, they can die. Let's see, what else do we want to know? And then that pretty much leads to the end of this chapter. Let's take another little break, and we'll carry on with the exam review. Hey everyone, welcome back. Let's continue exam 2 review with microbial genetics. Again, I'm going to be skipping some slides here, but please do learn everything. Know what a genome is, what chromosomes are. Definitely know what a gene is. A gene is a particular set of, it's a stretch of DNA that codes for a particular protein. So each gene codes for a protein, right? That's in the very, that's the essence of what a gene is. Obviously, it gets more complicated than that, but that's what I want you to know for now. Know the difference between genotype and phenotype. Genotype are the alleles or genes that organism possesses, whereas phenotype is what is actually seen. So a phenotype could be blue eyes, but the genotype would be the alleles blue eye, if that makes any sense. Let's keep going. Please know that nucleotides are the basic unit of DNA and RNA. Know what nucleotides are composed of. Nucleotides consist of a phosphate group. It's a 5-carbon sugar. In DNA, you've got deoxyribose, and you should know that in RNA, you have ribose, right? And then nitrogenous base. You should know that, again, these are the 3 components of a nucleotide, and a nucleotide is the basic subunit of DNA and RNA, the nucleic acids. And you should know this as well. You should know that, excuse me, you should know that DNA uses the nitrogenous bases, utilizes A's, G's, C's, and T's. And the only difference is that in RNA, instead of thymine, instead of T, you have uracil, U, okay? And you should know this. A's pair with T's with 2 hydrogen bonds. G's pair with C's with 3 hydrogen bonds. And what's the difference with RNA? The only difference in RNA would be A's pair with U's in RNA, okay? And you should know this for the exam as well, that 2 strands of DNA are held together by those hydrogen bonds, okay? Hydrogen bonds hold the 2 strands of DNA together. And that the DNA, the 2 strands are anti-parallel. They go in different directions. And by directions, I mean, you know how a strand of DNA goes 5' to 3'? It's directional. If you have 2 strands of DNA, one strand is going 5' to 3' in one direction, and the other strand is going 5' to 3' in the other direction. And notice how the nucleotides, nitrogenous bases, are held together by hydrogen bonds. That's very important to know. Okay. You should know these terms. Helicase, DNA polymerase. In fact, know all of these, but definitely know helicase, DNA polymerase. What do they do? DNA polymerase 3, primase, ligase, topoisomerase. Know that DNA is synthesized in a 5' to 3' direction with respect to the daughter strand. The daughter strand, the new strand, is being built in a 5' to 3' direction, and that would be the leading strand. The leading strand, by the way, the leading strand builds 5' to 3' towards the fork, right? Towards the replication fork where the helicase is unwinding the DNA essentially, right? On the lagging strand, you're still building 5' to 3', but that's going away from the fork. And that's why you end up with the Okazaki fragments. You should know that for the exam. Please know for the exam the difference between transcription and translation. Transcription is where you're making a copy of the gene. So the DNA is being used to synthesize complementary RNA called mRNA. And then in translation, the mRNA is being used by the ribosomes to produce proteins. All right, what else? Let us keep going. Oh, please know, again, what is the term mRNA? What are codons? You should know for the exam that each codon, or block of 3 letters, or triplet code, codes for a particular amino acid. So each triplet code, each codon, codes for an amino acid. Except for what? Except for the stop codons. Remember, TAG, TAA, TUA. Those are the stop codons. The stop codons do not code for a amino acid. All of the other codons code for an amino acid. What else should you know? Let's keep going. Okay, I'm just going through till I find something that I want to emphasize. Oh, inducible operons. Please know that with an inducible operon, I don't know if this is in your notes or if I've added it recently, but please know an inducible operon requires an inducer molecule to remove the repressor from the gene. And repressible operons are normally turned on, right? So remember those concepts. Very important here. All right, what else do I want you to know? Oh, I want you to know the term transformation, okay? I want you to know what transformation means. Remember, this is where small fragments of DNA can be introduced into a bacterium, and that bacterium can use that DNA in order to read those genes in order to gain certain functions. What else? Mutation. Definitely know the term mutation. Mutation are heritable changes in the genome. This is when you change the sequence of DNA, and that may or may not change the sequence of proteins, depending on whether it's a silent mutation or not. Okay, those were the things I wanted to... Oh, here, one last thing. The Ames test. Remember, the Ames test is a quick test to determine whether carcinogen is potent or not, right? So what you do is you add the carcinogenic chemical to a colony, or I should say to a spread plate, and then you look for conversions. You look for these bacteria that either gain an ability or lose an ability during the Ames test. And the more conversions there are, the more carcinogenic the bacterium is, because the more DNA was mutated. Okay, so what we're checking for with the Ames test is mutation rate, and that is indicative of what? It's indicative of carcinogenic potential of the chemicals. And that's it for this chapter. Let's go on to the last chapter, finish strong. Hey everyone, let's finish up this exam review with Chapter 11, Physical and Chemical Control of Microbes. let's see. Oh, please know these terms. Sterilization, as a nurse, as a dental hygienist, as any kind of healthcare professional, you need to know the difference between these terms. Otherwise, you're going to sound, like you don't know, like you have a grasp of what you're doing when you get into the clinical environment. So if you say something sterile, everyone's going to think, well, this is safe to use on a patient during a procedure. And that's a lot different than if you say something is disinfected. So sterilization, I want you to know that this is the destruction of all microbial life. It's the highest level of control. This is when you, for instance, put the surgical instruments into an autoclave and you kill everything, including viruses and, you should know, endospores. Endospores as well. Sterilization kills endospores and all other viable organisms. Disinfection is less stringent. It's less. For example, endospores can often survive disinfection. Antisepsis. This is where you use chemical agents on the skin. Decontamination is even less stringent. Okay, this is also known as sanitation. So if you were to go to a restaurant and you know the silverware they put out in front of you and the plates they put in front of you, that's all been sanitized. That's just reducing microbes to an acceptable level. Okay, let's see what else. Again, sterilization, process that destroys, removes all viable microorganisms, including viruses and, I want you to know, and endospores. Disinfection is slightly less stringent. Disinfection is not ideal for surgical environments, for instance. Okay, and obviously know these terms. I'm just flipping through to emphasize, as usual. Surfactants, you should know that a surfactant is a type of chemical, not a physical control method. It's chemical. You may need to know on the exam that surfactants are a chemical control method and not a physical control method. What else? Moist heat, dry heat, know those terms. An autoclave, you should know what an autoclave is. This is an instrument that increases the pressure and temperature such that it's effective at killing viable microorganisms, including those endospores. So when someone says something's been sterilized, oftentimes it's due to an autoclave session. Pasteurization, you should know what pasteurization is. Heating up a liquid below boiling, but sufficient to kill most microorganisms. Obviously know all these terms. Ionizing radiation versus non-ionizing radiation. Please know those terms. Okay. So for example, ionizing radiation, this would be like radioactive material, X-rays. Non-ionizing radiation, this would be, for example, UV, right? UV radiation. Those would be examples there. Let's see, what else? Filtration. Please know about filtration and what it's effective against. And let's see what else. All right, halogens include the elements fluorine, bromine, chlorine, iodine. Phenolics. Please know the concept of phenols. This was the first agent used by Lister, I believe, on his instruments in order to attempt to disinfect / sterilize them. What else? Alcohols. Please know about alcohols and what they, how effective they are. Hydrogen peroxide. Oh, and again, surfactants. Understand that surfactants are actually chemicals, not a physical control. Acids. and alkalis. Please know about them. And that's it. All right, so again, I hope in this video I was able to accomplish one main thing, and that is to give you certain concepts to really understand. If basically the way to think about it's like this: if you can't do anything else because you're stretched for time, or you just can't, at least know the concepts that I mentioned in this video. But ideally, if I was you, I would study all the material from the slides in order to do well on the exam. And I know you guys can do it. I hope this helped. And let me know if you have any questions for the exam by email. And I wish you luck, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.